Thank you for the introduction. So without further ado, let me start my presentation on biology in the era of foundational models. So they say you should start a good presentation with a promise. However, I wanted to start mine with stating the obvious, and that is the fact that cell biology is very complex. And in fact, it's more complex than anything we solved before with AI. And now I would like to tell you what makes it so complex so that you could really understand the motivation behind what we are working on. So first of all, cells are very dynamic and nonlinear systems, meaning that small changes in the input, like a small hotspot mutation, can lead to very, very complex changes in the output. And on top of that, cellular processes have massive number of interactions, which makes it very hard to model. So secondly, biological data is highly multimodal, meaning that if you really want to understand what's going on in a cell, you need to take all of those omics together and try to integrate them. And from the third side, the data is still very noisy, indirect, or obscured, meaning that we all have technical problems like batch effects and technical variations. And on top of that, omics technologies are destructive, meaning that your cells get destroyed during measurements. So, for example, if you do a single-cell perturbation-seq experiment, you cannot directly map a cell before perturbation and after perturbation. And now let me get to the question, what can we do about it? What can we do to understand biology a bit better? And here comes the perturbations. So it's like a golden engineering rule. If you want to understand how your system works, you should first break it and then try to build it up from scratch. And if you apply perturbations, such like chemical ones with a drug or genetic perturbations with a CRISPR technology, you could really try to learn the causal relationships between the genes, proteins, the pathways, and finally leading to the cellular functions. However, doing like a whole genome perturbation screening in the lab is still very costly, challenging, and time-consuming. And from the other hand, the data sets are quite limited to specific contexts of cell lines or drugs. So imagine you could just run the simulations on your computer for like 18,000 of genes, and from them you could choose 100 that are the top performing, the top promising, and just run the wet lab validation for them. So that's how we get to the concept of a virtual cell. So what I mean by a virtual cell? So by definition from the paper, How to Build a Virtual Cell with Artificial Intelligence, virtual cell is a model that tries to learn a universal representation of different cellular building blocks, such as DNA, RNA, and proteins, and it tries to integrate them across different scales, like molecular, cellular, and multicellular. So starting from the top, from the molecular scale, here we focus on the molecules that are in the central dogma of biology, so on DNA, RNA, and proteins. And these molecules can be easily represented by sequences of characters such as nucleotides or amino acids. So it makes them really easy to model with like textual-based models, like for example LLMs. And then moving on to the cellular scale, here we can represent a cell as a collection of these molecules. So each cell has a DNA sequence, RNA transcript, and protein quantities. And all of these molecules, they exist in a certain location with a certain quantity in a given point of time. So now we can try to integrate the universal representation learned from the previous scale with the spatial organization of the cell, using, for example, like single-cell RNA-seq data or single-cell ATAC-seq data. And from the modeling part, we can use transformers or autoencoders. And this way, we get to this universal cellular representation that knows its molecules, but also the spatial organization. And the final scale is the multicellular one, where we focus on how the cells interact with each other, how they communicate, and how they are forming tissues. As for the data here, we can use spatial profiling technologies, and as for the modeling part, we can use, for example, vision transformers for like spatial transcriptomics data or graph neural networks. So the core idea behind the concept is that you learn these universal representations from the previous scale and then you integrate them on the other one. And this way you could really like learn complex relationships and the hierarchy of your biological system. So I really like a metaphor here that these virtual cells, they're learning like a statistical shape of life. But now, why do we care about them? So I've been feeling lately that virtual cells have become like a buzzword because there's been this virtual cell challenge that I was talking about and a lot of publications coming on. But from the other side, I do believe, and we do believe as in genetics, that they could be like a holy grail of biology. So they should have three main capabilities. They should predict. So what happens if you knock out this gene? What happens if you add this drug to a given cell line? But they shouldn't only predict, they should explain you why. So for example, which pathways were altered, what genes were overexpressed and underexpressed. And only this way, if you predict and add the explainability, you could really discover new things, like discover novel therapeutic targets or drug combinations or new hypotheses. So the whole promise behind this is that with a good virtual cell model, we could shift the cell biology from being 90% computation, 90% experimental and 10% computational to the other way around. And now moving on to the second part of my presentation, I would like to explain to you what we are doing as Ingenics. So at Ingenics, we don't believe that one model can solve it all in biology. And we don't believe that there is one company that can gather all of the data and all of the expertise necessary to really tackle these challenges. And apart from that, so we are taking a radically different approach. We want to build like a model fusion platform that we called internally Octopus. So at the center of this model, or as this octopus head, we have our own proprietary AI model that is fusing like lower level models. And as these lower level models that we are calling tentacles, sometimes we have either first party models, open source models, or even our client's agent models. And we are integrating all that into like a GPT user experience so that you could really talk with our model. And the thing that's really important to us is the step-by-step reasoning, so that it could explain you why the answer behind the question, so that you could really see the evidence in a true world data. And this way, we want to simulate clinical efficacy and safety outcomes. So in short, there is clinical trials, because you probably all know that 90% of the drugs still fail in the clinic despite we have the AI revolution. So before the virtual cell challenge was born, that I will talk about, we are already trying to solve the perturbation prediction problem. So our goal was to test the ability of the octopus to perform like a casual biological reasoning between different scales. So from proteins to pathways to omics, phenotypes, and to the drug response. And for this reason, we chose a task to predict cancer cell line sensitivity after drug treatment using the GDSC dataset. So GDSC stands for Genomics of Drug Sensitivity in Cancer. It is composed of 10,000 observations, 700 cell lines, and 231 compounds. And our goal was like a binary classification. So you get a cell line and a drug, and you want to tell if this cell line will be sensitive or resistant to a given treatment. And so that we could really test if our model can generalize. So if it could generalize to the new context that it wasn't trained on, we wanted to select a difficult split. So if you have pairs of these cell lines and drugs, you can construct 3 of those splits, like leave pairs out, leave the whole cell lines out, and leave drugs out. And we've chosen this middle one, like leave cell lines out split, as it's the most complicated one, because we are excluding the whole biological context from the training data, and we're trying to generalize to this context. And our model performed really well on the GDSC task with just 4 agents. So as these tentacles, we have 4 different models. One for genes and proteins understanding, the second one for our internal model for molecular interactions predictions. The third one was for signaling pathways understanding built on top of the KEGG database. And the fourth one was for cell understanding built with DepMap omics data. And we significantly outperformed other known biomedical agents. You can see that on this GSC leaf cell line outspeed, we got the accuracy of 86%. Whereas, for example, the KX-Gemma model that was developed by Google DeepMind for therapeutic development, it got 51% accuracy, so that's like random accuracy. And as for the architecture design, we are training this in a DeepSeek-like manner. So we introduced 3 stages of training: massive pre-training, supervised fine-tuning, and reinforcement learning. And during different ablation studies, we proved utility of all of these different stages, and you can see how much they contributed to the overall accuracy. But as I told you before, the thing that we cared about mostly was if octopus can really provide reliable mechanistic hypothesis behind these answers. So now I wanted to show you an example. What happens if we ask the question, would viability or proliferation effect will be caused by treating a cell line T47D with a molecule called tamoxifen? So firstly, octopus head decides that it needs to gather the information about the cell line, about the drug and its targets, and then it can really answer the question. So that's like the whole head orchestrating the response. So then it calls one of the agents, the cell tool, and the cell tool correctly identifies that T47D is a breast cancer cell line with two very important mutations in TP53 and PIK3CA genes, being oncogenes and tumor suppressor genes, and also it's estrogen receptor positive. So then octopus calls the affinity and molecules tool, which says that the tamoxifen molecules, it targets estrogen receptor alpha, which drives like hormone-related proliferation signals. And finally, using the pathway tool, it can bring this knowledge together and say that since tamoxifen will modulate estrogen-driven pathways, and our cell line is estrogen receptor positive and has these two mutations, it depends on these pathways for growth. So it makes a hypothesis that the cell line will be sensitive to a given molecule, and that is in fact correct based on the experimental data from GDSC. So moving on to the final part of my presentation about the virtual cell challenge that I will shortly refer to as VCC. So Virtual Cell Challenge was organized by ARC Institute. ARC Institute is a non-profit research organization in the United States. It announced the competition late June, and it just ended this Monday, so it's still very fresh. And the task of this challenge was to predict like single cell expression distribution for unseen perturbation. But the real goal of this competition was to generalize. So learn from different perturbations on the same cell line and try to generalize to the unseen ones. And along with the goal of the competition, ARC Institute released its own dataset with 300 measurements, 300,000 measurements single-cell RNA-seq from human embryonic stem cell. So if you notice, a cell line, it's a really more complicated one as it's pluripotent, so it can differentiate into different states. So it's more hard to model the, for example, cancer cell lines. And also cancer cell lines are more present in public datasets, so that makes it far more challenging. And in this challenge, they've selected 300 CRISPR knockdown genetic perturbations, which they divided 150 for training, 50 for validation, and 100 for the test set. And it worked like this. So each day we were allowed to make one submission to the live validation leaderboard with our model's predictions, and then we could see the score. However, on the final test dataset that was available for the last week, we couldn't see the score. So we don't know how we performed on the test set. We will know early December when they will announce the results. Moving on to the metrics, the organizers selected 3 metrics to really measure things across different scales to ensure like biological interpretability and also the expressivity. The first metric was pretty easy. It was a mean absolute error between pseudobulk predictions and the targets. The second one was for differential gene expression. So it computed like a mean overlap between predicted and true differentially expressed genes. And the third one is probably the most complicated. It measured like relative differences between perturbations while computing the inverse normalized rank of Manhattan distance between predicted and target vectors. And you should note that even though the whole competition was on the single cell data, actually these 2 metrics were computed on pseudobulk vectors, and only the differential gene expression was computed on single cell populations. So when you enter a competition like a VCC, you should really take a moment and ask yourself a question: what does it mean for a model to be good? So what is the definition of success? And imagine that you've been working on some fancy deep learning model for a couple of months, and after some time you discover that it performs like slightly worse or just as good as a simple linear model, like linear regression or a mean baseline. And then you find this publication saying that deep learning models do not yet outperform simple linear baseline. So, oh well, that is not a great choice. But then you find this publication saying that they do outperform these uninformative baselines just on well-calibrated metrics. So now there's a lot of confusion going on in this field especially, and you should really choose carefully and wisely which one of these to trust. And I personally agree with the second one, and I will try to explain you why. But before that, I wanted to show you two histograms showing differences in the distributions between like the perturbed group and the control group in the gene expression. And I wanted to ask you which one of these two genes you think is a differentially expressed gene. You can say it out loud, you can say it in silence. I can wait.
So what was the question?
So the question was, would you say which one of these two genes is a differentially expressed gene? So where you see like significant differences in the distribution?
C.
So it was a tricky question, so sorry for that. I would say that none. However, the package released for the competition would say that B is a differentially expressed gene, while C is not. And that's like a very weird choice. And if you look closely at the titles, you can see why. And the reason is that log fold changes for these two genes are very tiny. So in the competition, they decided that some gene is a differentially expressed gene only based on the FDR threshold of the adjusted p-value and without any log2 fold changes filtering. And from my point of view, that's one of the biggest problems with these metrics, because if you're doing statistical tests for 80,000 genes times 50 perturbations with like Wilcoxon-Rank-Sum test, you're just inviting these false positives to come, and then you want like tiny shifts in the distribution. They seem differential, but they're really not. And here's a true differentially expressed gene with a good log2 fold change value. And there's a small disclaimer saying that I didn't really just cherry pick these examples on B and C. Actually, 56% of differentially expressed genes in the training data for the competition had log2 fold changes values lower than 0.25. So it made this metric really difficult, but also less meaningful from my point of view. And coming back to the question of metrics, we did a small experiment that we called like a 50-50 split experiment. So we took the training data, we split it in half, and we treated one part as a grand truth and the second part as the predicted perturbations. And then we ran the competition's evaluation suite, and we obtained this PDS and MAE metrics were pretty good. However, you can see that the DES metric, so this one for differential gene expression, was 0.42 with maximum of 1. So it showed us like the true upper bound of this metric that we can look for. And truly, none of these submissions for the leaderboard exceed like 0.48, I think. So that's a true upper bound. And coming back to the paper about well-calibrated metrics, the authors show that metrics such as MSE or Pearson correlation can be very easily hacked by predicting just the mean baseline or the control values. And imagine that you have like a weak perturbation where you have 18,000 genes and only 20 of them are really differential, are really altered. And if you put everywhere the control values, like the accuracy seems great, it's 99%, but truly you miss the biological signal because these 20 genes were the ones that we care about, were the ones where something changed. So that's why the authors proposed something called weighted MSE, where you put like higher weights on the important genes, for example, the differential one, and then you could really measure if your model learned something or not just hacked the metrics around. And about hacking the metrics, sadly, a lot of the focus of this competition went to hacking the metrics than to finding like biologically meaningful things. You can see, this is the top of the validation leaderboard. You can even see the name, leaderboard hacker. So, and you can see the MAE values, which are very high. While the MAE score of mean baseline, so just predicting the mean of all perturbations, was 0.02. So that means that these submissions were artificially boosted to boost the first two metrics, since for these metrics, the organizers decided that if you're performing worse than the baseline, the metrics is capped at 0, so there's no penalty if you have such a high value of MAE. But I do know that it's very hard to mess with MAE like that. And if you did, you, for example, multiplied everything by constant so that you could boost the first two metrics and it messed with the other one. Okay, so final part about the architecture. Unfortunately, it's hard for me to explain you the details about the model, but I would like to just show you the context. So before the competition, our institute released its own model called STATE for single-cell perturbation prediction. And it became like a go-to architecture for the competition. For us, we iterated quickly through different models like the state, flow matching, graph neural networks, NLP baselines, but we ended up with developing our own foundational model for expression modeling. So comparing it to the state model operates on cell tokens, so it doesn't have any internal gene level representation. We wanted to transpose this formulation and we wanted to represent the genes as tokens so that our model can really learn like gene-to-gene interaction and can be initialized from good pre-trained embeddings. Secondly, state model cannot incorporate bulk RNA-seq data, which we believe is really important. And also better quality and higher rate of biological context. So we are supporting bulk data, single-cell RNA-seq data, observational and also perturbational. And finally, state is like a hybrid of two model pipelines, which is more difficult to train, extend, and maintain. Whereas our model is like one model for different tasks, for perturbation prediction, for mask trans reconstruction, pathway masking, cell type classification, and many more to come. My final slide, so that's the architecture. We chose the perceiver architecture. It's a variant of transformer, which can handle like a long input context because we have a context of 19,000 genes. And I just wanted to show you one ablation where we were initializing gene embeddings from different embeddings, comparing random ASM, so embedding sequence model, DepMap CRISPR essentiality embeddings, and our own ASM PPI embeddings, which were fine-tuned on the protein-protein interaction task. And we showed that starting the model from these PPI pre-trained embeddings really improved the competition metrics, whereas in the state model, we didn't see any improvement with different embeddings, which showed us that our model can really learn from the starting point from the gene embeddings. So here I wanted to thank you for the attention, and also I wanted to thank you, the Nginx team, for collaborating on this challenge, and I think it was a great team effort and a lot of lessons learned. But before the questions, I had one of my personal favorite slides with a volcano plot. So throughout this challenge, I looked like thousands of volcano plots a day, and I wanted to show you what happens if you look at them too much. So that's my favorite one. Thank you.